this y-axis values. Now, clustering microarray data, genes and experimental samples are given as the row and column vectors of a gene expression data matrix. Clustering may be applied either to genes or experiments. So, why cluster? We cluster the genes, identify groups of possible co-regulated genes, for example, in conjunction with sequence data. Identify typical temporal or spatial gene expression patterns, that is, cell cycle data. Arrange a set of genes in a linear order that is at least not totally meaningless. We cluster the experiments or experimental samples. Quality control, detect experimental artifacts or bad hybridizations. Check whether samples are grouped according to the known categories. Then identify new class of biological samples, for example, tumor samples or subtypes. Now, here you can see the different groups or clusters are present. For example, lymph node proliferation, activated T cells, and then germinal B cells, then pan B cells. So you can see these are the different clusters, different subclusters. Here you can also see the clusters and subclusters. For these things, you just need to work on the threshold of these distances or the parameters which you have used for clustering. Now, generally, cluster analysis is based on two ingredients, that is distance measure or the algorithms. So quantification of dissimilarity of the objects, that is distance measure or similarity of the objects. Now, clustering algorithm, a procedure to group objects. Aim is small within cluster distances, large between cluster distances. So how to choose the number of clusters? There is no straightforward rule. It depends on what you want and when you feel that the solution has reached.